presentation, Ligand directed active tumor targeting PLGA nanoparticle for cancer chemotherapy. Gist of presentation: synthesis of this PLGA using ring opening polymerization of this glycolide and lactide. Then surface decoration of this PLGA surface with biotin ligand. This PLGA conjugated biotin ligand used to encapsulate irinotecan into this matrix, and these nanoparticles tested for different physicochemical in vitro and in vivo study as a proof of concept for CT26 inoculated colon cancer. In another work, LHRH conjugated PLGA-PEG matrix is formed, and this matrix is used for formulation of nanocapsule, which is loaded with docetaxel and quercetin, and which again characterized for different in vivo and in vitro studies as a proof of concept for PC3 induced prostate cancer in the animal model. PLGA is most successfully developed polymer in the drug delivery and tissue engineering application. This polymer is synthesized using two synthetic pathways. In dehydropolycondensation, this hydroxy group of lactic acid undergoes condensation reaction with the carboxylic group of the glycolic acid and results in the formation of this PLGA and water as a byproduct. It is very difficult to remove water from this high viscous solution of polymer, due to which there is a low molecular weight and broad polydispersity is observed for this polymer. But in case of ring opening polymerization, this cyclic trimer of lactide and glycolide in presence of different catalysts like some metal, nucleophilic catalyst, cationic catalyst undergoes ring opening polymerization to form this PLGA of high molecular weight and narrow polydispersity index. This PLGA polymer for drug delivery attracted considerable attention because it is biocompatible, as this lactide and glycolide obtained from sugarcane biodegradable. Due to this ester bond, this PLGA is degraded into the glycolic acid and lactic acid, which undergoes Krebs cycles and yields water and CO2 as a byproduct. This PLGA contains hydroxy and carboxylic group, and it is very easy to modify this surface. We can encapsulate hydrophilic and hydrophobic drug easily into this PLGA copolymer. When we see the market scenario, there are more than 25 products are approved by US FDA in the form of microparticles and implants for this PLGA carrier, and which are used for encapsulation of certain peptides, antimicrobial drugs, and anti-cancer drugs. And there are more than 50 drugs are in the clinical trial. So it is one of the most popular polymer in the 21st century. In the literature, Kanzi and co-worker studied the toxicity of most commonly used this catalyst in the polymerization reaction. And they observed that this catalyst shows toxicity in the human endothelial cells and Swiss 3T3 fibroblast cell. In another work, Zivkovic and co-worker studied the genotoxicity of this polymer in the human umbilical vein cells and mesenchymal stem cell. Although this catalyst is approved by US FDA based on the empirical safety data. In this empirical safety data, there is a heavy extraction and purification step to remove this stannous. And limit of final till in the product should be less than 20 ppm. So in our group, we have synthesized high molecular weight PLA using ring opening polymerization with the help of zinc proline, which is biocompatible catalyst, and zinc proline is also approved in the most of the immunological preparation. In the literature, calcium, zinc, aluminium, magnesium, and iron-based catalysts also reported for the synthesis of this PLGA polymer. And this polymer considered as biocompatible because of the biocompatible nature of this metal associated in the catalyst. In our work, we have synthesized different molecular weight of PLGA using 50-50% mole ratio of the glycolide and lactide monomer in presence of the zinc proline. Here we can see this is the seal ampule. Before reaction, this ampule is inserted into this glow box. Inside this glow box, we have to fill all reagents because these reagents are moisture sensitive. After that, we have to fill vacuum and seal in the glass bluing. And this reaction we have emerged in the sand bath. In sand bath, these ampules heated at higher temperature, and after polymerization, this polymer are dissolved in the chloroform and precipitated in the methanol. Then we will get this purified PLGA polymer. The mechanism of this reaction, there are different mechanisms associated with this ring opening polymerization like coordination insertion, monomer activated, carbene initiated, Brixlow intermediate. We have observed this one of the ligand of this zinc proline is replaced by this glycolide moiety. After that, it forms actual initiator. Then this initiator undergoes propagation with this lactide and results in the formation of PLGA. This study we have confirmed again with the DFT study, that is density functional theory, and these results are again agreement with the H1 and Maldi-Stock studies. We have synthesized different grade of this PLGA. We have used 50-50% of lactide and glycolide for L-lactide and varied EEG percentage from 5%, 10%, 15%, and 20%. For DL-lactide, we have varied the lactide from 60 to 90 mole ratio, and for lactide to glycolide, this 40, 30, 20, and 10. And we have observed that these polymers are not soluble. So these polymers are of no use because of the solubility problem. Then we have observed when we use L-lactide and the ratio of lactide is 75, 85, 65, and 60, whereas the remaining percentage is for glycolide. And for DL-lactide, when the ratio is 50-50, which is mostly used polymer in the drug delivery application, then lactide, that is DL-lactide is 60, 75, and 65, and the remaining percentage is glycolide. In this case, we have observed these polymers are soluble. Using this 50-50% mole ratio of lactide and glycolide, that is the feed ratio, we have carried out ring opening polymerization varying the reaction temperature, time, and monomer to initiator ratio. And we have observed at a low temperature, high reaction time, and higher monomer to initiator ratio, we have observed PLGA molecular weight around 87,000. This is the feed ratio, but actual incorporation of this lactide and glycolide monomer in this polymer is calculated using H1 NMR. These are other characterization like H1 NMR for confirmation of the formation of polymer, Maldi-Top for the end group analysis, viscosity to measure the viscosity, GPC to measure MW, Mn, and polydispersity of this polymer, thermogravimetric analysis to check the thermal degradation behavior and stability of this polymer at high temperature, wide angle X-ray diffraction, and DSC to check the nature of this polymer, whether it is amorphous or crystalline in nature, inductively coupled plasma mass spectroscopy in order to measure the concentration of zinc present in this polymer. After this, we have characterized this polymer using Bernoullian statistics, because if our polymer, that is PLGA, contains continuous sequence of lactide or glycolide, that is called as block polymer. and these block polymers are not soluble in the solution. In order to, your polymer should be have random sequence for its solubility. So this sequence we have confirmed using Bernoullian statistics. Again, we have deconvoluted this H1 NMR of this PLGA, and we have analyzed the sequence is random, and because of this random sequence, this PLGA is soluble. We have to use this PLGA polymer for drug delivery application. And when we see the history of modern delivery technology, it is around 60-year-old. In generation first, most of the oral and transdermal drug delivery products are successfully translated into clinical product because nature of problem is similar. It deals only with the physicochemical properties of the drug and polymer. In case of second generation, we can see in second generation, it is less productive for the development of product into clinical market because nature of problem is more complicated in the second generation. This direct delivery system fails to overcome the biological barrier. In this generation, drug delivery system use better control release because of the physicochemical properties of these drugs. In case of third generation of drug delivery, there is a need to overcome both physicochemical and biological barrier. And the prosperous feature of this drug delivery technology is depend on how this drug delivery product in the third generation interact with the human physiology. When we see the normal tissue, in the normal tissue, the endothelial cell are continuously lined with this pericyte and surrounded extracellular space with collagen fiber, fibroblast, macrophages, and there is a proper lymphatic vessel for maintenance of the fluid balance. But when we see tumor tissue, we can see these endothelial cells are discontinued. There is a high percentage of this collagen fiber. Due to this nature of the tumor tissue in passive targeting, when these nanoparticles comes into the circulation, nanoparticles comes into the circulation, due to this enhanced permeability, get accumulated at the tumor site, and as there is no lymphatic drainage in the tumor tissue, so it will take longer time for clearance from the circulation. But there is a most important disadvantage of the passive targeting is that it associated with the off targeting, and due to which there are side effects associated into the healthy tissues. In case of active targeting, we have to select particular ligand, which can be amino acid, vitamin, or any molecule, for which there are certain receptors like antigen or biomarker, which should be overexpressed into this tumor receptor. And in this case, there is a less side effect associated because of the proper active targeting to the receptor of the tumor. There are two types of strategies used in the active targeting. First one is towards the angiogenic endothelial cell. There are cells which are in the continuously rapid growing cell. So we have to use certain inhibitor of this angiogenesis. Then toward the tumor cell, there are certain transporter. This transporter is overexpressed in the tumor cell. As we have selected biotin, for biotin there is a sodium dependent multivitamin transport, and this transport is overexpressed in the cancer cell lines. So these nanoparticles are easily uptake by these cells due to this biotin. Due to this active targeting, there is improved cellular accumulation, uptake, and internalization. But there is a need to study the size, shape, and surface charge because it affects the particle distribution inside your body. If the size of nanoparticle is less than 6, then it is easily cleared by renal clearance. If it is above 200 nanometer, then it is uptake by the macrophages. Similarly for the liver, spleen, and lungs. We can see here biodistribution pattern for the lungs, liver, spleen, and kidneys varied for the size, shape, and charge of the nanoparticles. We have conjugated this biotin to this PLGA using Stiglitz esterification, that is the activation of this carboxy group using this DIPC, this nitrogen abstract, the hydrogen of this acid, then it become protonated, then this O minus attack to this carbon to balance this charge, and we will get this moiety. Again, this DMAP, dimethyl amino pyridine, which attack here and form stable intermediate. After that, this hydroxy group attack nucleophily over this DMAP and biotin moiety and results in the formation of this PLGA biotin conjugate. This conjugate is characterized using H1 NMR. This is H1 NMR of this PLGA. This is for biotin. At 12 ppm, there is a proton of this biotin. And after conjugation, there is a disappearance of this proton, which indicates conjugation of this biotin PLGA. This again we have confirmed with HABA iodine calorimetric estimation. This HABA iodine kit is available in the market. We have added our biotinylated PLGA to this solution. This biotin and iodine is the strongest non-covalent interaction in the biological system. That's why it replaced this HABA, that is hydroxyazobenzoic acid. Due to displacement of this HABA, there is a decrease in absorbance is recorded. And with the help of certain equation, we have calculated percent biotinylation of this PLGA nanoparticles. Then we have used this PLGA biotin conjugate for formulation of nanoparticle for irinotecan drug. Organic phase contain ICH class 3 solvent, that is acetone, with PLGA and irinotecan, which we have added into the aqueous phase. Aqueous phase contain surfactant. When this organic phase is added to aqueous phase, there is a displacement of the solvent, and you get these nanoparticles. These nanoparticles characterized by different physicochemical in vivo and in vitro studies. This is the characterization table for the PLGA loaded irinotecan and PLGA biotin loaded irinotecan nanoparticles. Hydrodynamic diameter, polydispersity, and zeta potential determined with the help of DLS. Encapsulation efficiency and loading efficiency calculated using spectrophotometric method. And we have observed at 4 degrees Celsius, these nanoparticles are stable up to 28 days. Surface morphology. Electron microscopy, and we have observed this nanoparticle has spherical morphology. Then with the help of selected area electron diffraction in same mode of TEM, we have observed this irinotecan is in crystalline phase. This irinotecan loaded PLGA nanoparticles and irinotecan loaded PLGA biotin nanoparticles are in the amorphous stage. Because when we see first image, there is a discontinuous ring of this circle, which indicates the crystalline diffraction of this irinotecan samples. Then atomic force microscopy study used to check the surface morphology. Here in 3D image, we can see the surface morphology. And in this line graph, we can see the spherical morphology and the size of these nanoparticles. We have carried out in vitro drug release study in the two medium, phosphate buffer at pH 7.5 and 50% butyl calc serum. And we have observed this irinotecan control release around 100% in the 24 hours. First release, sustained release. In the DC analysis, the sharp endothermic peak for this irinotecan is found to be absent in these irinotecan loaded nanoparticles, which indicates transformation of this crystalline irinotecan into the amorphous state. In wide-angle X-ray diffraction, irinotecan shows Lorentzian distribution due to crystalline nature, whereas irinotecan encapsulated nanoparticles shows amorphous nature because it indicates there is no diffraction of this peak. Then we have carried out in vitro cell viability using CT26 cell lines, and we have observed there is around IC50 value for this irinotecan loaded PLGA nanoparticle is 75 microgram per ml, whereas for irinotecan loaded PLGA biotin nanoparticle, it is less than 75 microgram. Here we can see untreated cells of CT26 cell lines. This is treatment of blank nanoparticles. This is for 75 microgram concentration of this irinotecan loaded PLGA and irinotecan loaded PLGA biotin nanoparticle. For this two concentration, we can see the cells are denucleated and there is a decrease in the population of this cell due to the toxicity associated with these nanoparticles. Then with the help of confocal microscopy, we have stained the cells with DAPI and nanoparticles loaded with this DIL dye. This dye used for the in vitro uptake of this nanoparticle. Then we have merged these two and we have observed for irinotecan loaded PLGA nanoparticle, there is a higher uptake of these nanoparticles in the cell. Then we have quantified this data with corrected total cell fluorescence and we have observed there is a higher intensity. Then we have to perform cell cycle analysis using propydium iodide staining. For control, we can see higher population of cell in the G0 to G1 phase. In case of irinotecan treatment, there is an increase in the cell population is observed for G2M phase, and which is retained for irinotecan loaded PLGA and irinotecan loaded PLGA biotin nanoparticles, which indicates these nanoparticles act on the G2M phase of the cell cycles. Then we have to confirm by which pathway these nanoparticles are internalized into the cell. We have treated this CT26 cell lines with certain inhibitors at 4 degrees Celsius. At 4 degrees Celsius, most of the metabolic processes of the cells are stopped. Sodium azide, which is inhibitor of the active transport, amphotericin, which inhibit the movement and uptake. Then cells with a nanoparticle, and we have observed at 4 degrees Celsius and sodium azide treatment, there is a no cellular uptake, which indicates this is the active transport pathway responsible for uptake of these nanoparticles into the cell. Then we have confirmed in vitro biodistribution study using DIR loaded nanoparticle. We have developed tumor of around 30 to 40 mm³ into the right flange of this mice. After that, we have given injection into the tail vein, and we have monitored fluorescence with the help of IVS instrument up to 7 to 2 hours. And we have observed there is an improved accumulation of nanoparticle at the tumor site. We can see here. Then after excise of this tumor from this mice, after sacrifice, we have collected different organ and we have quantified the fluorescence intensity. And we have observed there is a highest intensity is observed for liver, spleen, followed by tumor. And when we compare for tumor, there is a higher intensity is observed for diloaded with the biotin because of the active targeting of the biotin. Then biodistribution studies of the irinotecan performed with the help of HPLC using fluorescence detector. Here we have used 3 groups. One to sacrifice at 1 hour, 4 hour, and 24 hours, and we have quantified concentration of the irinotecan into the liver, lungs, tumor, and these different organs. Initially, at 1 hour, there is a higher concentration of this irinotecan is observed in the lungs, but after 4 and 24 hours, there is a decrease in the concentration is reported. Whereas for tumor, as the time proceeds, the nanoparticles start accumulating at the tumor site, and there is an increase in the drug concentration is. Then we have performed tumor site retention study. These diloaded nanoparticles are injected into the tail vein of these mice, and we have observed these nanoparticles are retained up to 9 days at the tumor site. In delay tumor growth study, the study performed using biopsy mice inoculated with the CT26 cell lines, and when this tumor is reached up to 50 mm³, we have started therapy. We can see these vertical lines indicate the day of injection, 8, 10, 11, 13, and 15 days. We have given 30 mg/kg dose for this irinotecan and equivalent nanoparticles. And tumor size measure with the help of Vernier caliber. And we have observed there is a delay in the tumor growth is observed. This control group and this treatment group. After sacrifice of these mice, we have collected this tumor, and there is a difference in the weight of this tumor. In body weight measurement, we have observed there is no change in the body weight of this animal, which indicates the non-toxicity of these nanoparticles. Similar for the organ weight, there is no change in the organ weight. In immunological study, we have to check how these nanoparticles interact with the immunological system. For this MDSC, Miller-derived suppressor cell, OXP3 and CD4 to CD8, which we have calculated from the spleen of these mice, and we have observed there is no statistically significant difference for this parameter, indicates there is no interaction of these nanoparticles with immunological system. Then we have to marker study, but there is a significant change for the neutrophils observed. There is a decrease in the irinotecan concentration is observed for irinotecan treatment, conjugated this PLGA with LHRH. First, we have activated this carboxy group with this EDCNHS coupling. Then this amino group of this PEG is conjugated to this carboxy group, and which is again conjugated with the LHRH, which contain amino group. This LHRH receptor are overexpressed in the prostate cancer. We have loaded docetaxel and quercetin into the nanocapsule of this PLGA conjugated LHRH using this solvent diffusion followed by polymer precipitation, and we can see drug is encapsulated into the oil core and shell is surrounded for this polymer. Transmission electron microscopy, we can see the core shell morphology of these nanoparticles. And qualitative cellular uptake. This qualitative uptake we have performed in PC3 cells in which LHRH receptor are overexpressed. We have loaded Nile red into this nanocapsule. Nucleus of cell is stained with H dye. Then PKH is stained with the cell, and we have overlaid the spectra. Here we have observed these are the PLGA, this is PLGA PEG, and this is PLGA conjugated LHRH nanoparticles. We have observed there is an increase in the cellular uptake as compared to control. When we quantify this, for this LHRH conjugated nanoparticle loaded with Nile red. Then in vitro cell viability study, we have observed there is a decrease in the IC50 value for this LHRH conjugated nanoparticles, and which are loaded with combinational drug, that is docetaxel and quercetin, as a LHRH conjugated nanocapsules, there is a improved accumulation at the tumor site. Again, we have carried out delay tumor growth study for this pancreatic cancer. This cancer using different groups, significant delay in the tumor growth. This is the tumor inhibition rate. In summary, we have synthesized two active targeting ligand nanoparticles. In first case with a biotin for colon cancer, and in second case with LHRH targeted nanocapsule for prostate cancer, and characterized as a proof of concept for colon cancer and cancer. These are my few list of publications. I would like to thank Dr. Sohan Chitlange, Principal, Dr. DY Patil Institute of Pharmaceutical Science and Research, for giving me opportunity to work in this organization. My research guide, Dr. Baijantimala Gharnaik, Senior Principal Scientist, Polymer Science and Engineering, NCL. Collaborators, Dr. Gopal Kundu, former Director, National Center for Cell Science, Dr. Niti Sharma, Symbiosis School of Biological Science, Department of Pharmaceutics for support and continuous guidance. Thank you. If you have any question, you can ask.
Thank you, sir, for your wonderful session. on topic ligand directed active tumor targeting PLGA nanoparticles for cancer chemotherapy. Am I audible? Am I audible to all of you?
Yes.
Yes,
Yeah.
ma'am.
Okay. Few questions. There are few questions. Sir, can you please explain again cell viability assay? Giram sir.
Yes, madam. In cell viability assay,
Please, will you please show the slide?
okay, yes, these are cell viability assay. Here we can see this is free. In order to study your nanoparticles in cell viability, you have to seed particular count of cell like 10,000, 7,000 cell on the petri dish. After that proper growth of cell, we have to add particular concentration of the nanoparticles or drug into that cells. After that, you have to remove the medium from cell and you have to add MTT dyes. After addition of MTT dyes, that is cell viability assay, we will come to know how many percentage of the cells are in active phase, in metabolic phase, and with the help of UV, you can measure the concentration of these cells and your control. It is clear?
Yes, sir. The next question has come that again, will you please elaborate in detail that about multi-targeting nanoparticle system or dual targeting nanoparticle system? Hello.
Here, in dual targeting system, we have used in dual targeting, we have encapsulated two drugs, that is docetaxel, which inhibit microtubulin polymerization, and second one is the quercetin. It causes sequential block of this cancer cell, and due to which there is a better therapeutic effect is observed. This is dual targeting. And in chat box, there is a question from Dr. Sushil sir. How you determine receptor density on tumor surface? There are certain formulas. With the help of these, we can determine the receptor density, but we have not calculated the estimation of receptor density.
There are a few questions in chat box. One more question has arisen.
Yes.
How do I troubleshoot the solubility problem of PLGA nanoparticle loaded with protein and prepared by double emulsion method?
Solubility problem of PLGA loaded with protein, and it is problem of solubility of your protein or solubility of PLGA. If it is the problem of solubility, you have to solubilize your protein with the selection of proper solvent, because this protein is denatured in the organic solvents. So for that, you have to check the compatibility of your protein. It is clear, sir?
Giram sir?
Yes, madam.
Am I audible?
Yes, madam. Yes.
Okay, okay, sir. Sorry for disturbance in between. Will you please tell how to increase the delivery of given drug to specific target site or targeting ligands?
How to improve targeting of nanoparticles?
How to increase the delivery of given drug to specific target site or targeting ligands?
For that, we have to select particular ligand, and this ligand is overexpressed into particular cancer. In our case, we know this biotin is uptake into cell by multivitamin sodium transporter. It is uptake into cell via multisodium vitamin transporter. So we have selected this biotin as a based on our cancer which we are to study. We can select this ligand. There is a list of ligands available in the literature for particular, because these cancer cells are rapidly growing and there are certain biomarkers which they are overexpressed.